Good morning, students. Today our topic is about clinical biochemistry, and in this we are going to discuss about role of enzymes and hormones in blood glucose levels. We all know that blood glucose level is most important for maintenance of a healthy human life because today the most important and dangerous problem is diabetes. It is due to the loss of maintenance of a loss of or imbalance of blood glucose concentration. So what are the enzymes and what are the hormones that are exactly useful in maintenance of blood glucose levels and to maintain a healthy life, and what are the conditions these enzymes and hormones are not acting in diabetes condition. By this video, I hope I am going to clear the how the blood glucose concentration is maintained in our body and how to regulate the diabetic people to make the normal blood glucose concentration. So, first of all, what is glucose homeostasis? It is the maintenance of blood glucose level within the normal range. The name itself indicates that homeostasis means maintaining of the balance. So we are going to maintain the balance of blood glucose within the normal range. The normal range of blood glucose concentration is 120 mg per 100 ml. Normal blood glucose concentration is 120 mg per 100 ml. And in the fasting condition, it is from 90 to 110 mg per 100 ml. So, the blood glucose level must be maintained within the narrow limits of 70 to 100. So, this is the approximate value of to maintenance of in postprandial or in the fasting condition. To maintain this level, that is 70 to 100 mg, our enzymes and our hormones are working continuously to maintain this blood glucose concentration. How they are maintaining this blood glucose concentration, what are the roles they are going to play, is the topic we are going to learn today. So before going to the topic, we have to know that why the nature has chosen that the every human body, that is one to chosen that 120 mg per 100 ml, that a blood glucose concentration. Why should be it 120? Why should not be it 150? Why should not be it 180? Why nature had chosen only 120 per mg per 100 ml, that is blood glucose, normal blood glucose concentration? Means it being 120 mg per 100 ml blood glucose concentration at the normal level, the blood's osmolarity, the blood's viscosity, and the amount of requirement of the glucose by the tissues and cells is properly maintained. That is the main reason why we are going to maintain the blood glucose concentration at normal level. So if the all the condition, if it is maintained at 120 or 70 to 100 mg per dl approximate narrow limits of concentration, our all the functions like maintenance of blood glucose, uptaking of glucose by the tissues, and maintaining the proper osmolarity and maintenance of proper viscosities happen, then we leads to a healthy life. That is the main reason, that is why the nature has chosen that where our blood glucose concentration is 70 to 100 mg per dl. So normal blood glucose level during the fasting condition, that is before eating anything, 70 to 110 mg per dl. Early morning before you are going to eat anything. That condition is called fasting condition. In that condition, the blood glucose concentration is 70 to 110 mg per dl. In postprandial blood glucose level, that means after you are taking off your breakfast, after you are taking off your meals, anything, that is called as postprandial blood glucose level. So that also be tested after you have taken the breakfast or meal. After you have taken a breakfast, you have to wait for 1.5 to 2 hours. Then only the postprandial blood glucose level is analyzed, and in that condition, it should be to 120 to 140 mg. Why you have to wait for 2 hours in postprandial condition means when you are going to eat the food, there is abnormal increase of the blood glucose concentration, and this abnormal level of increase of blood glucose concentration comes to normal level in 1.5 hour to 2 hours. But in diabetic people, it is not coming in that meantime. That's why they are not maintaining the 120 to 140 mg per dl. So in normal people, that is fasting condition is 70 to 110 mg per dl. Dl means deciliter, nothing but the 100 ml. And in postprandial condition, 120 to 140 mg per dl condition. And above and below, the above level is considered as abnormal, nothing but we are calling it as the diabetic condition. Here are another two words known as hyperglycemia. Levels below the normal range. Levels above the normal range. That is, if it is above the 120 to 140 mg, we can consider it as a hyperglycemic condition. And hyperglycemia means that is blood present in glucose. That's why we are calling it as a hyperglycemia. That is, glycemia means glucose in the blood. That's why we are calling it as a glycemia. And hypoglycemia, that is levels below the normal range, that is they are less than the 120 to 140 mg per dl. So 120 to 140 range should be maintained. Above condition is called as hyperglycemic condition, and below condition is called as hypoglycemic condition. In hypoglycemic condition, if there is no proper amount of glucose in blood, your normal functionings get altered and get dysfunction. That's why we should not be hyperglycemic and we should not be hypoglycemic. So we have to maintain this 120 to 140 mg blood glucose concentration every time. So we are telling that we have to maintain the blood glucose concentration at 120 to 140 mg. But how this blood glucose concentration is maintained? What are the conditions? What are the functions? What are the metabolisms that are happening to maintain this blood glucose concentration? So will take two conditions. First condition is, if you are having the more amount of glucose, that is you are taking the large amount of glucose in a normal condition, if you are not diabetic, what will happen means you are eating more and more food, there is abnormal increase of the blood glucose concentration, but our system is very active that it should not be make over amount of blood glucose concentration. To remove this, our enzymes and hormones and act like to make the blood glucose concentration in the normal level. For that, when heavy amount of blood glucose concentration is increased, what will happen? It uses the glucose and goes for the oxidation. That here oxidation means oxidation of glucose, nothing but the glycolysis. That is, it is meant for the energy, the first condition. And in second condition, if there is more excess amount of glucose, then it is oxidized. Some amount is oxidized. Then and again there is more amount of blood glucose concentration. What will happen in that condition? Glycogenesis. What happened? glycogenesis, that is synthesis of glycogen. It takes place in the liver. So excess amount of glucose is converted into the glycogen when there is a high blood glucose concentration. And still there is a large amount of blood glucose concentration. What will happen? That is, after filling of the oxidation, after happening of the glycogenesis, and the excess amount of blood glucose is converted into the lipid, and that process is called as lipogenesis. That's why if we are eating more, we are increasing the blood glucose concentration more, what will happen? It is converted into the lipid and stored in the form of fat, nothing but triacyl glycerol. So this is the condition where blood glucose concentration is in heavy way. And now consider the second condition, that is, if there is less amount of blood glucose concentration than this condition, there is less amount of blood glucose concentration than this condition, what will happen to maintain it? Our enzymes and hormones are acted and make the other metabolic pathways to maintain it. The pathways include first one, glycosenolysis. That is, in previously we have discussed that if there is more amount of glucose, glycogenesis is happen. If there is less amount of glucose, glycosenolysis is happen. That is, the already formed glycogen in the liver gets break up and come into the blood to maintain this concentration. Later, our diet, after digestion and absorption, if there is any digestion and absorption molecules is present, which is came into the blood glucose as early as possible to maintain this blood glucose concentration. And in third condition, gluconeogenesis, that is, mostly gluconeogenesis is done from non-carbohydrate sources like amino acids, lactic acid and other. So its aim is to maintain the blood glucose concentration of 70 to 110 mg when it is get decreased. So these two conditions makes with collaboration and cooperationally and maintain the 70 to 110 mg per dl concentration of blood glucose. So here I am giving the clear picture how this is maintained at organ level by considering the first condition. By considering the first condition, that is stimulus, blood glucose level rises, you have eaten more amount of glucose, blood glucose level get risen. In that condition, beta, when there is a blood glucose concentration is more, there is a secretion of beta cells of pancreas. That is, in pancreas there is a beta cells which will release the insulin into the blood. And there, when the insulin came into the blood, its action is making the glucose to take up by the body cells. And still there is more amount of glucose, it is converted into the glycogen. And by doing these two conditions, blood glucose level declines and it maintains the homeostasis of blood glucose concentration. This is the first condition. And in the second condition, if there is less amount of blood glucose, that is by coming, it is not maintained this level, it is decreasing less than this condition, then what will happen? Again pancreas will show that, that is pancreas alpha cells of pancreas release a new hormone called glucagon. So both the hormones are secreted by the pancreas. If there is more amount of glucose, insulin is secreted. If there is less amount of glucose, glucagon is secreted. And the glucagon action is, its action also to maintain this glucose concentration. How? By breaking the liver glycogen and releasing the blood glucose into the blood and maintaining this blood glucose concentration. So both the two actions are vice versa, and these two hormones will act and maintain the blood glucose concentration. So in our topic, we are already told in the title topic that maintenance of blood glucose by hormones and enzymes. So we have discussed about the hormones. So here is the another clear picture of how this blood glucose concentration is maintained. I will explain again. So this is the high blood glucose. When there is a high blood glucose, pancreas stimulate insulin, that is beta cells, and peripheral tissue stay cell takes an insulin signal to take the glucose by the peripheral tissues from the blood, and the excess glucose is taken by the tissues and normal blood glucose concentration is maintained. If there is less amount of blood glucose, again pancreas gets stimulated and pancreas alpha cells release glucagon and it breaks the glycogen present in the liver and release this blood glucose to normal blood glucose. In this way, it is maintaining the blood glucose concentration. Okay, I hope clear. Next. So to maintaining the this homeostasis, what is the what homeostasis? It's not a water homeostasis, it's a glucose homeostasis. To maintaining this homeostasis, there are mainly three factors are playing a major role. First one is metabolic process. We have already discussed that this metabolic process include glycogenesis and glycogenolysis. That is glycogenesis and glycogenolysis. That is, glycogenesis means glycogen synthesis, glycogenolysis means glycogen breakdown. So when there is a more amount of glucose, glycogenesis. When there is a less amount of glucose, glycogenolysis. In this form, it is maintaining the metabolic. By maintaining the metabolic process, we are maintaining the homeostasis of blood glucose concentration. To maintain this blood glucose process, what are the hormones playing? This is insulin and glucagon. Insulin is the hormone released when there is a high amount of blood glucose, and glucagon is released when there is a less amount of blood glucose. And in the renal mechanism, it plays important role. In ordinary conditions, there is no net release of blood glucose in the urine, but if you are having more amount of blood glucose concentration, it is continuously increasing. There is some sort of glucose also be excreted through the urine to maintain the proper blood glucose concentration. Here the fundamental regulatory mechanisms. Yes, clear that as blood sugar tends to increase, there is secretion of insulin, glycogenesis pathway is accelerated, synthesis of glycogen is happened, and utilization of glucose by the tissues is increased. As the utilization of glucose by tissues increase, it means there is more amount of glycolysis takes in place to give the energy. As blood sugar tends to decrease, there is a increase of insulin, secretion of insulin. Insulin decrease ratio of insulin glucocorticoids and growth hormone and increase production of glucose mainly by gluconeogenesis and glycogenolysis. Utilization of glucose by tissues is decreased. So I hope in this both condition, that is what is exactly happening, what is the role of insulin. I am again clear there. As the blood sugar tends to increase, there is if there is increase in concentration of blood glucose, there is increase in secretion of insulin. If there is more amount of secretion of insulin, what happen? The whole glucose is converted into glycogen via glycogenesis, and utilization of glucose by tissues is increased. As there is less amount of sugar, as blood sugar tends to decrease, that is insulin is no needed, that is decrease secretion of insulin and increase the production of glucose and utilization of glucose by the tissue. So here the another regulating mechanism, that is in the postprandial state, that means after your meals. Actually, here we are exactly telling that what is exactly happening in the whole process to maintain this homeostasis is, for example, liver contains glycogen. Liver contains glycogen. And this glycogen is breakdown, that is glycogenolysis is happened to give the glucose. And this glucose, when this condition is happening, when there is a less amount of glucose concentration, the liver glycogen is breakdown and glycogenolysis happen, and this glucose is utilized by the brain, and this glucose is utilized by our muscles, and this glucose is utilized by our erythrocytes. If there is large amount of glucose, what will happen? Pancreas will secrete, alpha cell secrete glucagon, and this glucagon enters into the portal vein, and it will increase the synthesis of glycogen by using the glycogenesis. And to maintain this blood glucose concentration, there are large number of, that is alanine, lactate, which are non-carbohydrate sources. That is, these are involved in the synthesis of gluconeogenesis to maintain the blood glucose concentration. This is the whole picture we are going to understand by using homeostasis of glucose. Here I explained here a simple chart diagram. That is, if it is high blood glucose concentration, if it is high blood glucose concentration, signals to release insulin, which may decrease the blood glucose concentration. If there is low blood glucose concentration, signals to release glucagon and high blood glucose. Here you are seeing that there are our title topic, that is role of enzymes and hormones, is get clear at the end of this topic, in the way that insulin and glucagon are the hormones involved to maintain the blood homeostasis, and glycogenesis and glycogenolysis are also be the two metabolic pathways involved in the blood glucose homeostasis. Now by coming to the, we have insulin. Insulin is the first hormone to be isolated. This is the first hormone to be isolated from all the hormones available today, and purified, that is extracted, crystallized, and that is um, its structure is properly established by having the two chains A and B and 51, a total of 51 amino acids, and it is first hormone to be artificially synthesized. Insulin secretion is associated with energy abundance. Insulin plays an important role in storing excess energy. That is, if there is more amount of blood glucose, then insulin secrete, and it is getting, that is transferring this whole excess amount of glucose to store in the liver as glycogen. In the case of excess carbohydrate, causes them to store it as glycogen mainly in the liver and muscles. As the excess carbohydrates that cannot be stored as glycogen are converted under the stimulus of insulin into fats and stored in the adipose tissue. If the condition is like that, there is large amount of carbohydrate which is to be stored as glycogen, and excess amount is present more than the glycogen to be stored, is converted into the fats and stored in the adipose tissue. That's why most people, more eating persons get obese condition due to the conversion of these glucose into the fats and stored in the adipose tissue. Here I will explain how the mechanism is done. Insulin stimulates extrahepatic uptake of glucose from the blood and inhibits glycogenolysis in extrahepatic cells and conversely stimulates glycogenesis. Here, the picture is if there is increase of if there is secretion of insulin, that is when insulin will secrete, when it should not be, when there is a high amount of blood glucose concentration. If the high amount of blood glucose concentration is there, what is the need to happen the glycogenolysis? There is no need. That's why what insulin will done means it stimulate to synthesize glycogenesis by inhibiting glycogenolysis. As a glucose enter hepatocytes, it binds to inhibits glycogen phosphorylase activity. That is, there are two enzymes in the glycogenesis and glycogenolysis. What are those two enzymes mean? For the glycogenesis, glycogen synthase is the main important enzyme, and for glycogenolysis, glycogen phosphorylase is more important. Here, insulin secreted means there is large amount of glucose in the blood. For that, there is an increase of glycogen synthesis means there is activation of glycogen synthase. If the glycogen synthase is activating, alternatively, glycogen phosphorylase should be inactive. Why means glycogen phosphorylase what it will do? It is increase the blood glucose concentration. Already there is a high amount of blood glucose concentration. For that reason only insulin is coming. So we don't need this further blood glucose concentration to be happen. That's why glycogen phosphorylase should be inhibited. When blood glucose levels are low, the liver does not compete with the other tissues for glucose since the extrahepatic uptake of glucose is stimulated in response to insulin, as already we discussed. When blood glucose levels are high, extrahepatic needs are satisfied and the liver takes up glucose for conversion into glycogen for future needs. Already we have discussed. And how this mechanism is operated by the insulin. Like the receptors for other protein hormones, as you already know that hormones goes and bind to the receptors only. Hormones goes and bind to the receptor. In the same way, insulin also be having the receptor. Here, insulin is a group 2 hormone. Group 2 hormones are nothing but the protein hormones, which will bind to the surface receptors only. Group 1 hormones will bind to the, they can internalize into the DNA region at the hormone responsive element. So we are not going to bother about it. We are discussing about the insulin receptor, which is a protein hormone, which is a group 2 hormone. For that reason, insulin goes and binds to its surface receptor. Like the receptors for other protein hormones, the receptor of insulin is embedded in the plasma membrane. That is, being it is a surface receptor, the receptor is present on the plasma membrane. The insulin receptor is composed of two alpha subunits and two beta subunits linked by a disulfide bonds. So it is formed of what? Receptor is formed of four chains, two alpha, two beta chains. Two alpha chain subunits and two beta chain subunits are linked by each other using the disulfide bonds. The alpha chains are entirely extracellular. That means if it is the surface receptor, the two alpha chains are, if it is a surface receptor, two alpha chains are at the upper surface. And how's insulin binding domains? While the linked beta chains penetrate to the plasma membrane. If it is a surface membrane, that is beta chains are embedded in the surface. Above are the alpha chains, below are the beta chains. Here it is the clear picture that is showing the alpha and beta chains. These two are the alpha subunits and these two are the beta subunits. So this is the plasma membrane. So they are embedded in the plasma membrane, beta subunits, and alpha subunits are present on the extracellular surface. And this is the hormone binding domain, and beta subunits having the ATP binding thyrokinase domains to carry forward the signal through the G protein. So insulin is produced by beta cells of the pancreas, and it is a small protein with 51 amino acids and they have a molecular weight of 5808. It is composed of two polypeptide chains connected to each other by disulfide bond. So it is a formed of two chains, that is 51 amino acids are divided into the 21 amino acids and 30 amino acids. So there is a chain and B chain which is combined to form the 51 amino acids, and this A and B are linked by disulfide bond. So here the positive and negative actions of insulin. Here you can see that hypoglycemic hormone insulin, how it will, that is, if there is more amount of glucose, insulin is secreted to form the lipogenesis. It makes the lipid, that is formation of lipids. If there is more amount of glucose concentration, insulin is secreted to form glycogen synthesis. And if there is more amount of glucose, insulin is secreted to uptake of glucose in a muscle and adipose tissue. If there is more amount of blood glucose concentration, insulin increase the glycolysis. And if there is more amount of blood glucose concentration, insulin is secreted to form protein synthesis. And at the same time it is activating, then at the same time it is inhibiting also. It increases the, that is, if there is a synthesis, so we don't need the glycogen breakdown. There is a protein synthesis, that is, we don't need the protein catabolism. There is uptake of glucose in the muscle and adipose tissue. There is we don't need the gluconeogenesis. There is more amount of lipid formation. There is we don't need. That is, one is activating, one is inhibiting. One is activating, one is inhibiting. In this way, the insulin maintain the homeostasis and the regulation to maintain the blood glucose concentration. These are the some enzymes and action of hormones, how they are the blood glucose concentration is. And hyperglycemic hormones are glucagon, epinephrine, glucocorticoids, adenocorticotropic hormone, growth hormone, thyroxine. So these are all hyperglycemic hormones. Hyperglycemic means if there is less amount of blood glucose concentration, these all hormones maintain the blood glucose concentration, that is, increase the blood glucose concentration. But insulin is anti to these all hormones. These are all increasing the blood glucose concentration, it is decreasing the blood glucose concentration. And the hyperglycemic hormone, the most important one, is glucagon secreted by the alpha cells of the pancreas. It increases the blood glucose concentration, promotes glycogenolysis, and if it is promotes glycogenolysis means it inhibits the glycogen synthesis. So it in insulin activates the glycolysis, it inhibits the glycolysis. Why means it is want to maintain the blood glucose concentration first, later the energy source. So and enhance the gluconeogenesis. And glucagon is a linear peptide of 29 amino acid synthesized as the inactive proglucon and later proteolytically processed within alpha cells of pancreatic cells and get active glucagon. So glucagon first of all secreted as proglucon, later is activated as glucagon. So glucagon also be a group 2 hormone, and it is also going to goes and binds to the surface receptor hormone only. So that's why its action is also be by at the surface level, and its secondary messenger is CAMP. That is, in on the surface, if glucagon will come and act from the surface, its action is taken below the plasma membrane by the CAMP, which acts as a secondary messenger. So here epinephrine, adrenaline. So when you are more excited means you want more amount of energy. If you want more amount of energy means, then increase in the indirectly epinephrine and adrenaline will also be increased blood glucose concentration, promotes glycogenolysis, enhance glucogenesis. Why they are increasing the blood glucose concentration? That is, when you are get excited, that means you are consuming more amount of energy. That's why the most of the glucose is going into for the energy purpose, but there is a decrease in the blood glucose concentration. To maintain it blood glucose concentration, epinephrine indirectly increases the blood glucose, indirectly increases the glycogenesis, and enhance the glucogenesis. In the same way, glucocorticoids action also as a hyperglycemic hormone. Thank you.